Hello, my dear students. Welcome to IKSR chemistry classes. In this video, we will have an overview of nucleic acids, which are very important biomolecules. Let us start our discussion with the term heredity. Every generation of each and every species exhibits similarities in properties with its ancestors in a number of ways. For example, a son resembles his parents in many aspects. It has been observed that the nucleus of a living cell is responsible for this transmission of inherent characters. It means the nucleus of a living cell is responsible for the transmission of inherent characteristics from one generation to another generation. The transmission of characteristics from one generation to the next generation of a species is called heredity. Let us now see three terms: cell, nucleus, and chromosomes. It is the cell of human being. It contains nucleus in the center. This nucleus of the cell of human being contains chromosomes. It is obvious that chromosomes occur in pairs in the nucleus. There are overall 23 pairs of chromosomes in the nucleus. The central part of the cell is called nucleus, and nucleus consists of chromosomes. Now the question arises, what are nucleic acids? The particles present in the nucleus of the cell responsible for heredity are called chromosomes. It means chromosomes are responsible for the transmission of characteristics from one generation to the next generation of a species. The chromosomes are made up of proteins and another type of biomolecules which are called nucleic acids. It means chromosomes are responsible for the transmission of characteristics from one generation to next generation of a species. That is, chromosomes are responsible for heredity, and chromosomes are made up of proteins and another type of biomolecules which are called nucleic acids. The nucleic acids are mainly of two types, and these are deoxyribonucleic acid, which is abbreviated as DNA, and ribonucleic acid, which is abbreviated as RNA. Thus, nucleic acids are of two types, DNA and RNA. The nucleic acids are long chain polymers of nucleotides, and therefore they are also called polynucleotides. It is obvious that nucleic acids are polymers whose monomers are nucleotides, and this is why nucleic acids are called polynucleotides. The complete hydrolysis of nucleic acids, that is DNA or RNA produces mainly three types of chemical species, and these are a pentose sugar, that is a carbohydrate containing 5 carbon atoms, phosphoric acid, that is H3PO4, and a mixture of nitrogen containing heterocyclic compounds which are called nucleobases. The complete hydrolysis of nucleic acids, that is DNA or RNA, produces three types of compounds. First one is a pentose sugar, the second one is phosphoric acid, and the third one is a mixture of nitrogen containing heterocyclic compounds which are called nucleobases. It means DNA or RNA consists of, are made up of these three components: a pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compounds. The sugar moiety present in DNA molecules is beta D2-deoxyribose. The pentose sugar, the sugar moiety present in DNA molecules is beta D2-deoxyribose, while on the other hand, the sugar moiety present in RNA molecules is beta D-ribose. Let us see now structures of beta D2-deoxyribose and beta D-ribose, which are pentose sugars and are present in nucleic acids. Beta D-ribose and beta D2-deoxyribose. This is the Haworth projection formula of beta D-ribose, which is present in RNA. It is a carbohydrate containing 5 carbon atoms. It has 5-membered heterocyclic ring, and this is the structure for beta D2-deoxyribose. It is pentose sugar present in DNA. Let us look upon these two structures of pentose sugar. The OH group present at C2. In the pentose sugar, beta D-ribose of RNA is replaced by hydrogen atom in pentose sugar of DNA. In DNA, the OH group present at carbon number 2 is replaced by hydrogen atom. That is, this oxygen atom has been removed. This is why the pentose sugar of DNA, is called beta D2-deoxyribose. It means the oxygen atom present at carbon number 2 has been removed. Bases of DNA. DNA contains 4 bases as follows. Adenine, it is abbreviated as A. Guanine, it is abbreviated as G. Cytosine, it is abbreviated as C. And thymine. These 4 bases are present in DNA. It may be noted that all these bases are heterocyclic compounds containing nitrogen atom in the ring, and these are the bases present in DNA. Now, the bases present in RNA. RNA also contains 4 heterocyclic nitrogenous bases. The first 3 bases, that is adenine, guanine, and cytosine of RNA, are the same as in DNA. but the fourth one is uracil instead of thymine. It means RNA contains four bases. The first three, adenine, guanine, and cytosine of RNA, are the same as in DNA, but the fourth base of RNA is uracil instead of thymine, and the formula of uracil is given here. It is obvious that RNA contains four bases: adenine, guanine, cytosine, and uracil. Let us see the term nucleobases. The bases adenine, cytosine, guanine, thymine, and uracil obtained from nucleic acids are called nucleobases. These five bases are obtained by the hydrolysis of nucleic acids, and hence these are called nucleobases. Out of these five nucleobases, cytosine, thymine, and uracil contain pyrimidine moiety, and hence they are called pyrimidines. The three bases, cytosine, thymine, and uracil, have six-membered pyrimidine moiety, and therefore these three nucleobases, cytosine, thymine, and uracil, are called pyrimidines because all these three have pyrimidine ring. And purines. The nucleobases adenine and guanine contain purine moiety, and hence they are called purines. It is adenine and it is guanine, and these two have purine moiety, and hence adenine and guanine are called purines. Let us see the term nucleoside, a chemical species formed by the attachment of the nucleobase to 1' position of pentose sugar, beta-D-ribose or B-D-2-deoxyribose, if contains a nucleobase at 1' position, the chemical species formed obtained is called a nucleoside. This is a nucleoside which contains a nucleobase attached at 1' position of pentose sugar. This pentose sugar is beta-D-ribose, and it contains a nucleobase attached to 1' position of this pentose sugar, and hence it is nothing but a nucleoside. In nucleosides, the sugar carbon atoms are numbered as 1', 2', 3', 4', and 5'. In order to distinguish these positions from the positions in bases, the numbering in bases is performed as 1, 2, 3, 4, etc., while the numbering in the pentose sugar is performed as 1', 2', 3', 4', and 5', in order to distinguish the numbering in the base and in the pentose sugar. Example of a nucleoside: it is beta-D2-deoxyribose. It is the pentose sugar present in DNA, and it is adenine, a nucleobase present in both RNA and DNA. If this sugar contains nucleobase at 1' position, the chemical species obtained is called a nucleoside. Here the nucleoside is called deoxyadenosine. This is beta D2-deoxyribose, and this is adenine moiety, and the nucleoside is called deoxyadenosine. And the next term is nucleotide. When the nucleoside is linked to phosphoric acid or phosphate group at 5' position of sugar moiety, we get a nucleotide. It means if a pentose sugar contains nucleobase at 1' position and phosphate or phosphoric acid group at 5' position, the chemical species obtained is called a nucleotide. An example of nucleotide is given here. It is a pentose sugar which is attached to base at 1' position and phosphate group at 5' position, and hence it is a nucleotide. Example of a nucleotide. This is beta D-ribose sugar moiety. Beta D-ribose sugar moiety. It contains adenine nucleobase at 1' position and 3 triphosphate group at 5' position, and hence it is adenosine triphosphate. This is adenosine and it has triphosphate group at carbon number 5' and therefore it is called adenosine triphosphate, which is abbreviated as ATP, which is a well-known biomolecule, and it is a nucleotide. Dinucleotide. If two nucleotide units are joined together by phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar, we get a dinucleotide. Let us see it again. If two nucleotide units are joined together by phosphodiester linkage between this phosphodiester linkage is formed between 5' and 3' carbon atoms of pentose sugar, and thus we get a dinucleotide. An example of dinucleotide is given below. It is a nucleotide. It is another nucleotide, and these two nucleotide units are attached together between 5' and 3' positions through phosphodiester linkage. This linkage is called phosphodiester linkage. Two nucleotide units are attached together between 5' and 3' positions through phosphodiester linkage, and thus we get a dinucleotide. And polynucleotide, that is nucleic acid. In nucleic acids, a number of nucleotide units are attached together through phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar, and hence they are also called polynucleotides. Polynucleotides, that is nucleic acids, are long chain polymers of nucleotides in which nucleotide units are attached together by phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar. It can be represented as follows. Sugar is attached to base at 1' position. This is another sugar attached to having nucleobase at 1' position, and these two sugars are attached together at 3' and 5' carbon atoms biphosphodiester linkage. This group is repeated several times in the nucleic acid molecule and it is a polynucleotide. Thus, nucleic acids are polynucleotides having a number of nucleotide units attached together between 5' and 3' carbon atoms by phosphodiester linkage. And nucleic acids are present in the chromosomes which are responsible for the transmission of characteristics from one generation to the next generation of a species. That is, nucleic acids are responsible for the transmission of characters or heredity. Thank you for watching the video with patience. Please like, share and subscribe it. Thank you again.